Good afternoon and welcome to Cadlam Cymru revision sessions. This session will focus on AS biology and will be presented by Mr. Cobb from Ysgol Uwchradd Aberteifi. The session will last around 45 minutes where the teacher will go through the relevant subject content. If you have any questions, please use the question and answer section and we will endeavour to answer your question during the session. The session will be recorded with the recording and any relevant resources uploaded to the e-sgol website in the Cadlam Cymru area. Over to you.
Right, good afternoon everyone. I hope you're all well. This is what we're looking at. We're going to try and cover today during this session. We'll get through as much as we can. We're going to look at the basics of enzymes. There are certain key terms, key definitions that we're going to need. And then we're going to jump on to, as soon as we can, we'll look at the factors affecting enzyme reactions. So we'll look at the different aspects that can affect enzyme reactions and try and understand them as best as we can. And that'll be interspersed with some past paper questions to give us an idea of the type of question you can expect. So without further ado, let's get on to, and we'll try and do this as quick as we can, let's get on to the essentials. So look, I'm saying that there are certain aspects of this topic where you really need a definition. Okay, you need to know, this is your bedrock. Your understanding of the topic is fundamental here, because really almost certainly what you're going to be asked to do is to apply this information. So here's let's work through these terms first of all, and then we'll tie that into the diagram as well. But I'm hoping this is all old news. So a catalyst we know is a substance which speeds up the rate of a chemical reaction. We could change the name catalyst to an enzyme and then put the definition as a substance which speeds up the rate of a biological reaction. But a catalyst is just, it speeds up reactions. You're familiar with the terms substrate and active site, and if we look at the diagram here, we can see an enzyme, we can see the shape of the active site, and we can see how the substrate fits quite nicely into that shape of the active site there. So the substrate, the molecule upon which the enzyme works, and we say this substrate is complementary to the active site, meaning it fits. We can't say it's got the same shape, we say it fits, or it's complementary is the perfect word. So there's the active site, the portion of the enzyme molecule into which a substrate fits. And what have we got here? When they join, we have an enzyme substrate complex. So it's defined as the structure form when an enzyme and substrate associate with one another. Now, the principle here is that, look, it's a reversible reaction. So when the enzyme substrate complex forms, actually, there's two things that could happen here. It could go back the other way if conditions are not favorable. It could go back the other way and it could then dissociate and reform the enzyme and the substrate. But really what we're trying to do is get as much of this happening, the enzyme and the product being formed, or the product being formed, and regenerate an enzyme that's able to go and do its job again. The term specific or specificity is a term we use a lot with enzymes, and it's just this principle that each enzyme catalyzes one reaction alone. Okay, so that we say that enzymes are specific. It's not an uncommon question. Three more things to look at. So the first one is the activation energy. Okay, so again, question that's asked quite a lot. Activation energy, the energy needed to start a chemical reaction off. Now, I mentioned just now, if I go back a slide and say, look, if conditions are favorable, we can get the formation of the product, okay, and regeneration of the enzyme. Well, when we say are conditions favorable, we're saying, is there enough energy for the enzyme substrate complex to overcome this activation energy? So you've got two diagrams here, which just trying to explain exactly what enzymes or catalysts do. So we got reactants on the left-hand side on this pink one, we got reactions on the left and products on the right. And in order for the reactants to form these more stable products here, they've got to overcome this little energy barrier. Okay, that's what we call the activation energy. So you've got to be an input of energy first to overcome this barrier so that those reactants can form products. Now remember, what enzymes do is they speed up reactions. So the reactants will naturally, eventually turn into products, but this can take anything from weeks, months, years, or longer. What enzymes do is they lower this bump of energy, this energy barrier, and therefore make it easier for these reactants to overcome that energy barrier. So by reducing that activation energy, it'll make it easier for those reactants to become products. And as a result, then it happens an awful lot quicker. Now, the way in which that the enzyme and the substrate combine, there's two hypotheses as to how that works. There's a lock and key hypothesis, which is one from GCSE. So the idea that you have a substrates here that fit perfectly or complementary fit into the enzyme molecule and then form those products. Okay, but look at the shape of the substrates, look at the shape of the active site there. In this case, you're seeing two substrates being divided into a product. It's good to see that actually, because quite often when we show enzyme reactions, it's usually we show it with one substrate because it's actually quite easy. It's simpler to show that. But with the in the other type, the other hypothesis is the induced fit. Look at the shape of the active site here of the enzyme. Can you see it's not quite the same as the substrate there? And therefore, what actually happens is when the substrate approaches the enzyme, then the active site will sort of mold, will change shape just ever so slightly. And then it will look, once they bind, once the enzyme substrate complex is formed, it will bind and look an awful lot like the lock and key hypothesis at that stage. But prior to that, when you look at the free enzyme, the shape of the active site looks similar to the substrate, but it doesn't look perfectly complementary. So those are the essentials, really. That's the key terms and definitions you need. And my advice to you on these is, look, you just need to learn these ones. Okay, there are certain things in biology where to make your life easier, you just need to learn the definitions. And whilst that is hard work, learning definitions like that, you will find that with certain concepts, whilst you may understand it, coming out with a key wording in an exam is tricky. So whilst you sort of, you know what you're saying, but you're not quite sure what the right words are. Yeah, that sort of thing is where you need a definition. Right, okay, so I've done that quite quickly because I'm aware that what we want to really focus on is the applications of this, because that's the thing that people find most tricky. But before we do that, let's have a little look at an application of this. So I'll give you 20 seconds, 30 seconds to have a look at this. And then I will go through the answers. Okay. Right, so it's quite nice really this. Look, they're asking about what's represented by X on the diagram. Well, look, it's a very typical graph that we've just seen of reactants versus products, and X there is our standard activation energy, so should be quite comfortable with that one. It says enzymes are biological catalysts. Draw a line on the graph to show how the energy changes would take place if an enzyme was present. Well, we just draw a lower activation energy. Anything lower than this peak there would be just about fine. And then it says this, look, succinate dehydrogenase is an enzyme found in mitochondria and is involved in respiration. The enzyme catalyzes the conversion of succinate into fumarate. Using your knowledge of enzyme structure, explain why this is the only reaction succinate dehydrogenase can catalyze. So this is asking about the specificity of enzymes. There's 2 marks, so we know we're not going to just be able to say specific, we're going to have to give detail. So let's look what it's got here. So it says that the active site has a specific shape and succinate has a complementary shape to that active site. And it says therefore fits or binds or bonds into the active site. So look, it's one of those questions where you'd be fishing for the marks and thinking about what is it I can say that would give me the chance to get the marks here. Really, it's a case of trying to hit those key terms. Talk about active site, talk about complementary, talk about specific is what you're after. Okay, right, let's move on. This is what we're going to look at now. We're going to try and look at as many of these, like I say, as we can, the effect of these different factors in or how they affect an enzyme catalyzed reaction. So let's deal with temperature and pH first, and we'll start with temperature then. So this is our standard graph. We're going to see how temperature affects the rate of reaction. Okay, and I've got a question on here which says describe and explain the effect that temperature has on enzyme activity. And what I'm going to do is a little walkthrough here. We're going to talk about section A, the increase, and then we're going to talk about B, the decrease. And we're going to try and hit as many key terms as we can. Okay, so the first thing, and I've shown it in red here, what you see in red is the description. So let's just work on a little bit of exam technique here. Describe is say what you see, what is going on with the graph. So as temperature increases, the rate of reaction increases. That's my description. Okay. And explain. So now I'm going to say why that's happening. So I'm going to do it I'm going to combine it. But it's actually quite a good idea. Certainly if this is something you don't always get quite right, you could sometimes confuse the two to keep them separate. So the describe would be as temperature increases, rate of reaction increases for this section here, this section A. And then we'll say this is because, and now we're into explaining, because the molecules, okay, the enzyme molecules, the enzyme and the substrate have more kinetic energy or they're moving faster. You can say either is a mark for either. Okay, so you can say either one of those. That means there's going to be more successful collisions. Now, a successful collision is a collision in which the enzyme and the substrate combine. You know, they bind, they fit, and so on. But it results in the formation of a product. So the enzyme substrate combine, form an enzyme substrate complex. If it goes on to form a product, that's a successful collision. If it doesn't, it was an unsuccessful collision. Okay, so if we have more kinetic energy, we've been putting more energy into the system, so there will be more successful collisions. Now, you can also say this, there are more enzyme substrate complexes formed. So those are usually the same marking point. You can say either. They don't mean the same thing. But if you had more enzyme substrate complexes formed, it's likely you would have more successful collisions. But like I say, they are usually the same marking point. And therefore, so rate of reaction increases. So there in red, I've got my description. As temp increases, rate of reaction increases. And in black, I've got this is why. Well, what about over here? So once we get past the optimum here, so as the temperature increases past the optimum, I'm going to talk about denaturing here. Okay, so be careful. We don't say at the optimum or at whatever it is, 40 degrees. We get denaturing. It's as it goes past the optimum, okay, or above 40 degrees or whatever it is we might have on our graph. So hydrogen bonds break. Okay, key, we've got to talk about hydrogen bonds. Remember, enzymes are proteins. They're held together by hydrogen, hydrophobic, disulfide, and ionic bonds. It's the hydrogen bonds that break when the temperature gets too high. That causes a change in the shape of the active site, and therefore it is no longer complementary to the substrate. That means the substrate no longer fits, and we can sum that all up as saying the enzyme denatures. It's not uncommon to see students talk about hydrogen bonds breaking, changing the shape of the active site, and say it's no longer complementary, it doesn't fit, and so on. And they forget to use the term denatured. You know, it's a key biological term, so we've got to make sure we include that. But quite common because they've done such a good job in the explanation, they forget actually the simple stuff. So, and then the rate of reaction decreases. So as temp increases past the optimum, rate of reaction decreases. There's our standard stuff, okay, that we're going to need to use shortly. Now, this graph and this knowledge is very, very common. I would say, well, they both are common, okay, how increase in temperature increases rate of reaction, and then the details of denaturing are both common. Detail needed, that you see on the screen, detail is needed. When it comes to pH, it's far less common. It's less common. I would say for every one question on pH, there's 8 to 10 questions on temperature. Okay, and it's probably a good thing because actually describing and explaining how pH affects enzyme activity is, I'm not sure I'd say it was more tricky, but it just, I always say this, whenever you describe a graph, you'll start with as whatever's on the bottom, the independent variable, as pH increases, the rate of reaction, so on. But I say in this case, we should always start in the middle. Okay, for pH, we say each enzyme has its own optimum pH. If we have a small deviation from the optimum, we get inactivation. Okay, so it reduces rate of reaction. Inactivation, in contrast to denaturation. So denaturation is a permanent change to the enzyme. Okay, you cannot get that back. So if you went in terms of temperature, if you went to really high temperatures and denatured the enzyme, and then you returned your temperature right back to 20 degrees or something, the enzyme still won't work. Okay, it's permanent. If you get inactivation, is where it's not working, but it's not a permanent thing. We actually can say that with temperature. It's like 0 degrees C. We might say, yeah, the enzyme is inactive, meaning that if we restore the temperature something more normal, or like body temperature or something, then yeah, it will work. So it's been inactive at 0 degrees, but it will still work. Well, the same thing applies here with pH. Small deviation from the optimum results in some inactivation. Now this is because, and this is where the difficulty lies, this is because this is quite high end here, meaning this is a rare question you're asked, and often chemists find this a lot easier to understand. But it's not a big deal. It's not something that appears that commonly, so it's not a huge problem. But if you do, if this does sit well with you, well, it doesn't cost you a great deal to remember it. So what am I talking about? As a result of, so why do we get this inactivation? Why do we get a drop in rate of reaction? Because the charges on the acid and base groups of those side chains change, they alter. And if you alter the charges, it's going to affect the ionic bonds. So ionic bonds are bonds between side chains or R groups with different charges, opposing charges. And if you alter those charges, then you're going to affect those ionic bonds. You break those ionic bonds. If you get a large deviation from the optimum, so here's our optimum here, small deviation, yeah, we get a drop off in rate of reaction. If you get a large deviation from the optimum, that reduces the rate of reaction further because they are then denatured. You have so many changes in those ionic bonds. There's so many ionic bonds that are broken that it becomes an irreversible change to the enzyme. Now, I'm not going to do any more on that because I could say it's quite rare that we see questions on pH. Okay, far better. Let's get on to this is the thing we really need to work on is focusing on graph work, and because you almost certainly when you have questions on enzymes, you're going to see a graph. The biggest bit of advice I can give, the best bit of advice I can give is first thing you do, check the axes. Okay, so really down the bottom on the x-axis, there's usually two things we can see: time or substrate concentration. Okay, now if you get tuned in to what the axes are telling you from the start, it gives you a better idea of what you can be expected to do. Okay, so rather than just learning shapes of different graphs, we need to look at what the graph is actually showing us. Now I just showed you a graph back there where you had pH on the bottom. Yeah, okay, that's a GCSE thing. And if I take you back again to temperature on the bottom, yeah, so I said, it's time, yeah, and then we see substrate concentration, but here are temperature and pH. Okay, you can see others, but most likely thing we're going to see is the time versus something like this. I'm saying the amount of product produced. I don't like to use the word amount. In fact, it's a general rule of never use the word amount, but we're talking in vague general terms here. So whilst I might have said the volume of oxygen produced or something like that, or the concentration of a given chemical being produced, that's fine, but I'm talking in general terms here because we're just talking about a general reaction. So I'm just using the general term of amount. So this is what I expect to see happen. As this takes some explaining to begin with, but this is going to be the bedrock of what we do later on. So as time increases, we can see the amount of product increases, but there comes a time where it levels off. At that point, all of the substrate is being converted into a product. We have reached the maximum amount of product that is possible, because at the point at which all the substrate is converted to product, there's nothing else that can happen anymore. We can't convert any more substrate to product because it's all being used up. So this line levels off and it will stay level and it will not change because effectively the reaction is over. We'll use this, we'll come back to this again and again. So it's worth keeping an eye on making sure we're happy with this graph. So I've now added this, let's say that's sort of low temp, but what would happen if you had it at the optimum temperature? The optimum temperature, what's going to happen? Well, look, here's a line. How's it different? It's steeper. The steepness corresponds to the rate of reaction. So reaction is going to happen faster at a higher temperature or closer to the optimum temperature. We are still going to get to this maximum level. We're not going to go any higher than that because that's not possible. we will still get all substrate converted to product, but it will happen sooner. So that point at which all the substrate is converted to the product happens here, as opposed to being further along on this graph over here. That's what happens when you go to the optimum temperature, rate of reaction is increased, so all that substrate is converted to the product quicker. If we went to something hot, 80 degrees, we're going to have some denaturing. Well, no, the enzyme will denature completely. All those enzyme molecules will denature. So you see down the bottom here, you can't see it particularly well, but you see an orange line here that corresponds to what happens if you're using denatured enzyme. How much will a product will be produced? None. It will stay there. It will never get up to this max. It will never get anywhere near it because the enzyme is not working. It's denatured. So it's worth keeping an eye on that. We could do the same for pH. And if we take, look, there's our standard, we call it the optimum pH. What happens when we go to a lower pH or slight deviation from the optimum? We're going to have a drop in rate of reaction. Okay, but it's not going to be huge. There's our line. So what happens? Yep, as time increases, the amount of product produced increases because we've got a lower rate of reaction. But we will get there in the end. All that substrate will be converted to product eventually. It will just take longer. So if you look at the optimum pH, look at the point at which it reached that maximum product and drop a line down, we're over here somewhere, but with a slight deviation from the optimum, a reduced rate of reaction, where do we drop that line down? Much further on. So we've reduced the, by slight deviation from the optimum, it's a drop in rate of reaction, and therefore we're going to see this sort of graph when we look at it on the side, how much product is produced. If we did a large deviation from the optimum, it's denatured. So we're going to see the same thing as we saw on temperature, and we're going to see that line along the bottom there showing show that there's no substrate converted to product. Let's have a look at a little question. Okay, again, I'll give you 20 seconds. I want to try and get through as many of these things as possible. So let's see how we get on. 20 seconds, and then just so you familiarize yourself with the graph. So notice what I said there about amount being a word we shouldn't really use, but it's a vague general term. Look what we've got here now. It's the total mass of product formed. There we are. That's better. That's how it should be, more like a mass of product formed. Look what we've got here. Explain why the initial rate of reaction was slower at 30 degrees than 50 degrees. So this goes back to our temperature graph. Why is it that as you increase temperature, it increases the rate of reaction? It's all about successful collisions. And enzyme substrate complexes. This is what I said before. Look, we got a less, so at 30 degrees, we have less or low kinetic energy, or molecules are moving slower, which means we have fewer successful collisions or fewer enzyme substrate complexes. And that gets us our 2 marks. Very predictable question, actually. Okay, so worth being completely familiar with that. The only thing I'd say that's not that normal there, usually you can say either low kinetic energy or the molecules are moving slower in this case. And then explain the shape of the curves between 30 and 60 minutes at 50 degrees C. Okay, so this is what happened at 30 degrees C. You might say, well, this looks a bit, this is not what we've seen. Why is it leveling off sooner? Well, because in this case, what happened is, well, that enzyme and that substrate were added together, and then it was put in at 50 degrees C. So it just took a bit of time for those enzymes to denature. It's a really bad way to carry out an experiment, to be honest. If you're going to carry out an experiment at a particular temperature, you probably know that you need to get your enzyme and your substrate up to that temperature separately, and only then would you actually join them together. You know, would you pour one into the other, if you like. But okay, the important thing here is what we're getting, the key terms relating to denaturing. So you say the enzyme is denatured, or you can say, look, alteration in the 3D structure, breaking of hydrogen bonds, that's key. Active site is changed and the substrate cannot bind, or less or fewer enzyme substrate complexes formed. So standard stuff, and you know that links in very closely with what I described on how temperature affects rate of reaction. Right. Okay. Let's see if we can move on. So we've done temperature, we've done pH. Those are just an extension from GCSE. Let's look at how substrate concentration affects rate of reaction. So here's our standard graph we'd expect to see. Look, it's changed now, okay? Down the bottom now, I've got concentration of substrate and rate of reaction up the side. I'll say it again. Whenever you see a graph, look straight away at, right, what is it on the bottom? Is it time or is it concentration of substrate? It's a key routine to get into because it just tunes you into the correct graph that you're going to need to be trying to recall in your head. So here's the essentials. As substrate concentration increases, the rate of reaction will increase. So that makes sense because if there's more substrate, obviously the reaction can happen faster. But at high substrate concentrations, if you have a further increase in concentration, it doesn't affect the rate of reaction. Okay, so we don't have an increased effect on rate of reaction. So you might think it would just keep going up and keep going up and keep going up. It doesn't. Once we get to a certain point where concentration of substrate is relatively high, further increases in substrate concentration, it doesn't get any higher. So effectively, we seem to reach a maximum rate of reaction. Okay, the reaction cannot go any quicker than this. So let's explain what's going on here and why that's the case. Okay, so here's an overlay. Here's the graph that I just showed you. Okay, I've got 4 situations I'm going to walk you through here from bottom left up to top right then. So set it up, what's going on? I got an enzyme down here in blue, and I got, this is my key, enzymes in blue, substrates are in green. So let's go to a low substrate concentration. I've got, look, I'm talking about really low numbers here that are really just to try and make this point clear. I cannot imagine a situation where we have 5 enzymes on 1 substrate, but hey, we're just trying to make the point. So we're talking about really low substrate concentration. What we've got at this point is look at all these enzyme active sites that are available to carry out or to speed up a reaction. It means that they're sort of twiddling their thumbs, if you like, and they're ready. They're ready to go faster to make that reaction happen faster. So if we go up here and now we add 3 substrates, add 2 more substrates, we've got 3 in total. Yeah, we do see an increase in rate of reaction. Absolutely, because there's enzymes there ready to do the work. So there are still at this point, if we sort of assign 1 enzyme to 1 substrate, a very crude way of doing it, but okay, you get the idea. It leaves us with 2 enzyme molecules, which is sort of free, if you like. So we're just twiddling our thumbs again. We're ready to do some work. Then if we go up to a high level here, where I got 5 enzymes and 5 substrates, now they're all busy. Okay, now the enzymes are all occupied. The active sites are all occupied with substrate. So they're busy. They're all busy. They're all working. And therefore, if you go higher and you increase the substrate concentration further, it's not going to make a difference because all the enzyme active sites are occupied. Okay, when it gets to this plateau, we would say that all the enzyme active sites are occupied. Therefore, further increases in substrate have no effect on the rate of reaction. We can say also that at this sort of this phase where we've got the slope on the graph, we say the substrate concentration is limiting the rate of reaction. So the reason the reaction isn't going faster is because there isn't enough substrate there. Whereas at the top here, this plateau, at that section, it's the enzyme concentration that's the limiting factor. The reason that, look at this one on the right here, a D if you like on there. Can you see you've got way more substrates than enzymes? But it's not going any faster when there were only 5 substrates. Okay, you've got 12 here, we've got 5 there. That's because it's the enzymes that are stopping. There's enough substrate to make it go faster, but we haven't got enough enzyme to get to a higher rate of reaction. So we've reached our maximum rate. So that maximum rate, which I referred to back here, and we'll refer to again and again, that maximum rate that I referred to here is governed, all other things being equal, by the enzyme concentration. Okay, so if we want that to lift, we're going to need to increase the enzyme concentration. Right, okay, let's have a look at a question. Okay, again, I'm going to give you just 20 seconds to have a look at this, and then we'll move on. Okay, as always, you can pause these videos and have a go at these questions yourselves, and then look at it afterwards if you wish. But let's have a look. So the graph below shows the, well, do you know what? in the interest of time, let's just look at what we've got going on here. This is our standard graph. We've just seen it's concentration of succinate. So it tells us, right, we are talking here about concentration of substrate. Okay. The stem of the question we're not seeing here. It would have told you what the reaction is, what's going on. So succinate is the substrate, fumarate is the product. And so that's telling us the rate of fumarate production is the rate of product production or rate of reaction. What's the limiting factor? The region marked Y. Well, that's going to be our concentration of substrate, succinate. Use evidence from the graph to support your answer. Well, because look what's happening. As the concentration of substrate increases, the rate of reaction increases, which tells us that it must have been the substrate which was the limiting factor there. And then it says, what's the limit in the rate of the region marked Z? Well, now it's the enzyme concentration. So it's the concentration of succinate dehydrogenase enzyme. And how do you know? Because all of its active sites are occupied. Okay, and that at any given moment is just saying, yeah, we appreciate. Well, it's a if you take a specific point in time, we're going to look at see that all those active sites are occupied. Right. Okay. Substrate concentration done. Let's try and move on now to enzyme concentration. So enzyme concentration seems really straightforward, and it is really because more enzyme means substrate converts into the product quicker. As simple as that. This is a graph we virtually never see in exams because look, can you see we've got enzyme concentration on the bottom, and what did I say? Usually it's time or substrate concentration. We don't see enzyme concentration because it's just too logical, I suppose. It's too easy to do this one. But well, of course, the more enzyme you have, the faster the rate of reaction. Now, it does assume the concentration of substrate is in excess, or I say extreme excess. You cannot consider, when you see in this graph, you can't think, oh, what if the substrate runs out? No, don't think like that. Assume that there's so much more substrate that it's simply a case of, as I increase enzyme concentration, there is going to be substrate there to be catalyzed. So we see this linear relationship between enzyme concentration and rate of reaction. Actually, when it comes to enzyme concentration, this is far more likely to be the case as what we're going to see. Look, we've got time on the bottom again, amount of product produced again. So this is our time versus amount of product graph, as opposed to substrate concentration on the bottom and rate of reaction on the side. There's our standard. That's where all substrates converted to product. That's the maximum substrate, maximum product that can be produced. That's what happens if we have a low concentration of enzyme, or what happens when we have a high concentration of enzyme. What's that graph going to look like? This is similar to what we did with increasing temperature, or getting to the optimum temperature, because we're getting to, we're having a higher rate of reaction, and therefore we are going to get to the same concentration of a product produced, or amount of product produced in this case, but we will get there sooner. Okay, the point at which, how long did it take there? Right at the end of this graph, did we actually get to that maximum concentration or amount of product? Whereas with a high concentration of enzyme, the reaction will happen quicker. So we're more like the middle of the graph, that we get all substrate converted into product. So a bit of detail on that. High concentration, we get the same total amount of product produced, but that total will be achieved quicker. And that's because there's more enzyme active sites to do the work. It's quite important to be talking about active sites. The exam board like to see that, as opposed to just saying there's more enzyme. There's more enzyme active sites there to do the work. Right, or to catalyze the reaction would probably be better wording. Right, okay. I'm now going to move on here to inhibitors. Okay, we look at about 10 minutes to try and cover the inhibitors topic. So let's see how we get on. So, basics for inhibitors: they slow down reactions, and it's worth mentioning that you are almost certainly going to have questions on inhibitors. They are probably the most likely thing to be asked about. So enzymes is such a common question to see. It's usually a long question, and you could have a whole mix of questions you could be asked about within that enzymes section. And inhibitors is almost certainly going to be on there. Okay, so what do? They slow down the rate of reaction. And the way they do that is they interfere with the way the enzyme works. But okay, two types: a competitive inhibitor. So the details here are going to be important. It's quite common to be asked, what is meant by a competitive inhibitor? So how does competitive inhibitor works? Well, the word competitive is all about competing for the active site. So in order to do that, you can see the inhibitor So the diagram down here has got a similar shape to the substrate, this little sort of diamond shape here. And the idea is that they are both complementary to the active site of the enzyme, so they're both going to fit into it. And therefore, they are both competing for the active site of the enzyme. They're both trying to get in there. And it's all about who gets in there first. Whilst the inhibitor is in that active site, it means the enzyme will not be converting the substrate to product. So therefore, the rate of reaction is reduced. Now it's temporary, okay? It's reversible. The inhibitor will bind with the enzyme and then it will associate, then it will dissociate. But whenever that inhibitor is bound to the active site, the actual real substrate can't get in there. And therefore, it's slowing down the rate of reaction. Now, the other type is a non-competitive inhibitor. So a non-competitive inhibitor does not bind at the active site. It binds away from the active site. It is not competing, hence the non-competitive. It's not competing for the active site. So you can see the shape of the inhibitor is different to the substrate. Instead, it binds away from the active site. So you can see the shape of the inhibitor and the shape of this sometimes referred to as an allosteric site. Okay, the term's not important, as in you don't need to remember that, but just know it binds away from the active site. When it binds to that location, it also will change the shape of the active site. So binding, and you can see here, binding at this location on the enzyme causes the active site to change shape. And now it's no longer complementary to the substrate. So perhaps the most important thing for understanding how non-competitive inhibitors work, and the graphs we're going to see on this, is that a non-competitive inhibitor can bind at any time. It is independent of the substrate binding. It's not about who gets there first. It's simply about whether the inhibitor is bound to the enzyme or not. But it does not have to sort of check that the substrates, is the substrate bound to the active site? Doesn't matter. It's available. It can bind to the to this alternative, this other site at any time. Now, whilst it's bound, so it means the inhibitor can bind to the enzyme at any time, and it's not a race now to get to the enzyme first. Whilst it's bound, that reaction can't take place, and therefore what we are doing here, and I've underlined this and put it in red, so what they do is they effectively reduce enzyme concentration. Now that's different to reducing enzyme concentration, okay, because it does not change the number of enzyme molecules. But having a non-competitive inhibitor and having a proportion of enzyme bound with non-competitive inhibitor at any one time effectively reduces enzyme concentration, meaning it has the same effect as if you did reduce enzyme concentration. So I'll try and explain that with some diagrams in a moment, and we'll look at the graphs too. So let's have a look, first of all, at a very nice graph to begin with. It's time along the bottom and amount of product produced up the side. There's our standard. Okay, usual, all substrate converted to product. What happens when we had an inhibitor? With an inhibitor, it does not matter whether it's competitive or non-competitive. It slows down a reaction. So we got to be wary of this. That's the graph we're looking at. It's time versus amount of product produced. So an inhibitor, whether it's competitive or non-competitive, looks the same for this graph. The more, why is that? With an inhibitor present, reaction is slowed. Same total amount of product will be produced, but that total will be achieved slower, and we've seen that before. Now, substrate concentration versus rate of reaction is a different matter. So here's our standard without a competitive inhibitor. Now we're going to add a competitive inhibitor. We know that there's our maximum rate of reaction there. Okay, so it's rate of reaction, it's maximum, and we've seen this before. That's because at this point all active sites are occupied. Okay, that's what we've said before. With the competitive inhibitor, look what we've got. So now we can look at two different scenarios here. If we look at low substrate concentrations down here, we know that because we've got low substrate concentration, any inhibitor that's present has got a good chance of getting to the active site, and therefore the active sites will be occupied by an inhibitor, so the reaction will certainly be slowed. But at high substrate concentrations, now there's going to be so much substrate present that actually the inhibitor has so little chance of getting to the active site first that the effect of the inhibitor is negligible. It doesn't really have an effect anymore. So actually at very high substrate concentrations with a competitive inhibitor, the rate of the reaction is unaffected at very high substrate concentrations. Again, I'm going to show you this in a moment a little bit better at what's going on. But note the shape of the graph looks exactly the same. I'll just reverse it a little bit. If I go to that, look, the shape of the graph is the same, but these are different graphs. This is time versus amount of product. And now this is substrate concentration versus rate of reaction. So whilst they look the same, the explanation is very different. Let's show what's going on here. So look, here's low substrate concentration. So we haven't got much substrate, but we have got in it. So I got my substrate in green, my inhibitor in red. So we're using the same key as before, but this time I'm using my inhibitor. I've added an inhibitor which is in red. It's competitive because it looks like the substrate there. Now, therefore, look what's happening here. Low substrate concentration. Look, the inhibitor's got a good chance of getting there. It's got a good chance of getting to the active site. So it's going to slow the rate of reaction time down. At high substrate concentrations, okay, so first of all, I've started off as the same. Now let's add some substrate. Now look at the situation. Whilst it's still possible the inhibitor will get to the enzyme, it's very unlikely. There's so much substrate here that actually those enzymes are going to be working as hard as they possibly can all the time because they're all going to be occupied certainly, and the likelihood is that they're all going to be occupied with the substrate, and therefore at high substrate concentrations we are still going to get that maximum rate of reaction that we would have had without the inhibitor. So that rate of reaction is unaffected. That maximum rate of reaction is unaffected at high substrate concentrations. It's a tricky concept. It's commonly asked. Okay, the key thing here is that at high, so the effect of a competitive inhibitor can be overcome by increasing the substrate concentration significantly. Non-competitive inhibitor, remember, it effectively reduces enzyme concentration. There's without the inhibitor, it's substrate concentration versus rate of reaction. With a non-competitive inhibitor, now we see a difference. Okay, now we see a difference. We are not going to reach that maximum rate of reaction because we are effectively reducing enzyme concentration. We could get that same result if we reduced the enzyme concentration, but that's not what's happening here. We've added a non-competitive inhibitor. Why? Because a proportion of the enzyme will always be occupied by inhibitor, so effectively it reduces enzyme concentration. Here's that graph again. Here's I'm going to try and so this graph here on the right here is just showing actually that's what a graph looks like if you lowered the enzyme concentration. So if you looked at high versus low enzyme concentration, the graph looks the same. As to what's going on, look, this isn't perfect here because I've used the same. I've shown I've drawn the red inhibitor as looking like the competitive inhibitor. I mean, it looks like it's complementary to the active site, and we know that non-competitive inhibitors do not work like that. But for ease of showing this, I thought I'd put it on like that. So we know that at any one time, a proportion of the enzyme molecules will be bound by inhibitor. So I'm saying that, look, at any one time, let's say 2 out of the 5 are bound with an inhibitor. Effectively, they're out of action then. It won't be the same 2, okay? The inhibitor may bind to this enzyme and then dissociate and bind to another enzyme. But if we sort of set a precedent and sort of start then and say, at any one time, 2 out of the 5 will be bound by an inhibitor, we may as well just say, let's just forget those exist. And now we've got a situation where we've only got 3 enzymes. We haven't only got 3 enzyme molecules, we've got 5. But given that 2 of them are out of commission, because they're bound by an inhibitor, we have effectively reduced enzyme concentration. Now that's true whether you get low substrate concentration or high substrate concentration. The fact that we've got a higher substrate concentration does not make a difference. We've effectively gone down to 3 enzyme molecules, and therefore we are never going to be able to get up to that maximum rate of reaction that we had before because there are fewer enzymes to do the work there. It's a tricky concept. I would revert back to the explanation I gave, but I hope that as somewhat you can sort of cling on to, but I hope the diagram helps. Now, one last thing we'll look up before we finish. Quick question on here. I'm looking at time, so I'll give you a very quick 20 seconds here, and then you can always pause if you're rewatching this and you can have a look again. So that is a quick 20 seconds. So let's see how we go. Right, what is meant by inhibition? Where an enzyme's action is slowed down or stopped by another substance. Should be quite nice, straightforward question. Why is malonate a competitive inhibitor in here? Well, it has a similar shape to succinate, which is the substrate, and so it competes with succinate for the active site. Now we've got, look on here, we've got substrate concentration versus rate of reaction. That's the graph we're after, substrate versus rate of reaction. Here's our normal enzyme. A is our typical competitive inhibitor, B our non-competitive inhibitor. So have a look what we got here. Which line A or B would represent malonate? Well, it's competitive, so it's line A. Why? Because if we increase the substrate concentration, the effect of the inhibitor decreases, or we can reach that maximum rate at a very high substrate concentration. Non-competitive, such as heavy metal, why with the rate of reaction with non-competitive inhibitor does it not reach the uninhibited that of an uninhibited enzyme? Well, the inhibitor binds away from the active site. The fact that it says it's often permanent, that's actually a rare marking point that we see, and it's not relevant here. Is it not relevant for sort of, don't you learn that for the future? But it alters the shape of the active site, yeah, fine. And then it effectively reduces the enzyme concentration, or reduces the number of successful collisions or enzyme substrate complexes. Okay, it's a little bit of a whistle-stop tour of I'm going to leave this one. About what I'll do, I'll give this 2 seconds for you to do it yourselves, and then I'll put up the mark scheme on here. And we're going to see a similar thing actually to what we've had as well. So similar shape, it's complementary. There is competitive inhibitor again. It asked then about a graph, what you draw to show the rate of reaction when a malonate is present is similar to the question you've just seen. And there we go. So look, whistle stop tour, and I hope it was useful. I hope you can play it back if you need to and look at the key parts of that, or parts you struggled on. But I hope you found it useful, and good luck with your revision.